Thanks, Jerry. So it's been a long trip from Stanford since I've been in St. Louis now for a little over 5 years, but it's a slow journey. But anyway, thanks for having me here. So I'm going to talk today about the interplay of somatic and germline variants in MPN. And these are my disclosures, nothing specifically relevant to today's talk. So I know we have an sort of a mixed audience in terms of background, so I just want to start with some very basic terminologies, hopefully to keep everyone on the same page here for the talk. So you've heard these terms before, but just to be clear, so when we're talking about germline and somatic, so germline is something, in your germline, it's something that you're born with that could potentially be passed down to your descendants, and it should be present in every cell in the body, which is different from a somatic or acquired mutation, which again is acquired. It's only present in specific cell populations. It could be acquired through DNA damage, exposure to carcinogens, and as I'll mention briefly during the talk, really is also part of normal aging. We use the terms driver and passenger mutations to distinguish those mutations, the driver mutations that are thought to be pathogenic, that are actually contributing to a specific cancer, versus passenger mutations, which are sort of along for the ride. So those are mutations that occur, but they don't necessarily have any functional significance. And the term exome sequencing refers to specifically looking at the coding regions of all the genes versus whole genome sequencing, which covers the entire genome. And there are different reasons why you might want to use either of those technologies. Okay, so the sort of modern era of genetics in the MPNs really started in 2005 with the landmark discovery of the JAK2 V617F mutation in all three of the classic MPN subtypes. And we know that this JAK2 mutation is present in almost every patient with polycythemia vera, but only about 50 to 60% of patients with ET and myelofibrosis. And since this initial discovery, those of us in the field have pondered this sort of very basic question, which is, how can you have this singular mutation which, quote unquote, causes these three different diseases? Certainly, PV, ET, and myelofibrosis share many characteristics clinically, but they are certainly distinct in many ways. And to elaborate on this, and Jerry had some slides on this as well, but the JAK2 allele burden varies depending on the MPN subtype, and it's thought that that could contribute to some of the phenotypic differences that we see. So JAK2 allele burden tends to be higher in PV, lowest in ET, and then a little bit variable in myelofibrosis. So certainly that could be one aspect that contributes to the different clinical phenotypes. This is probably hard to read, but also it's well established that the JAK2 status within patients with essential thrombocythemia can result in specific differences in phenotype. So again, within ET, you have JAK2 positive and JAK2 negative patients, and those who are JAK2 positive tend to have a higher hemoglobin and a lower platelet count compared to their JAK2 negative counterparts. Also, as shown here, there is a correlation with a specific JAK2 allele burden. This is again within ET patients, again with higher hematocrit corresponding to higher JAK2 allele burden, lower platelet count, et cetera. So there's definitely an association there with essentially the percentage of cells expressing the JAK2 mutation. This is a little bit more theoretical, but could it perhaps be that the specific cell that acquires the JAK2 mutation, or the cell in which these diseases originate, could that in some way specify the MPN phenotype? And again, there's not really good data to support this specifically, but you could imagine that if the disease or where the mutation is acquired is in a true stem cell versus some cell that might be along the more committed progenitor line, that this could specify the phenotype. So for example, if you've got a progenitor cell that's more disposed to produce red blood cells, maybe that would give you more of a PV phenotype and vice versa. So again, not real good data for that specifically, but that certainly seems plausible. And germline variation. So we'll talk more about this coming up, but within mouse studies, this was sort of proposed a while back that this could be a factor, primarily because in the mouse models that were developed to study the effect of the JAK2 mutation, it was observed that the phenotype was different whether you used one particular strain, Black 6 versus BALB/C, specifically that a more of a myelofibrosis phenotype was seen when BALB/C was used as the host for the JAK2 mutation. So that, again, no specific data to necessarily say this is true, but perhaps there could be germline differences in the two mouse strains that would be responsible for giving you these slightly different phenotypes. So I've talked so far about the JAK2 mutation. We know it's highly prevalent in the different MPNs, but is that the only mutation? And we know that certainly is not the case. So within the JAK-STAT axis, in addition to the JAK2 V617F mutation, there's a separate group of mutations in exon 12 of JAK2, which are very rare but are known to be present in selected patients with polycythemia vera. Mutations in the TPA receptor MPL, also not particularly common, but are known to be present recurrently in patients with ET and myelofibrosis. And then work that was conducted by myself in collaboration with Jason Gottlieb, who will be one of the speakers at this meeting as well. We identified mutations in LINK, which is a negative regulator of the JAK-STAT signaling axis in occasional patients, in particular with ET and myelofibrosis. So there are multiple members of the JAK-STAT signaling pathway that can be mutated in these diseases to the point where it might be true that the vast majority, if not all patients, have some genetic mechanism to activate JAK-STAT signaling. And this became a little bit confusing with the discovery of mutations in calreticulin, or CalR, several years ago, which are now known to also be highly recurrent, particularly in those patients with ET and myelofibrosis. Confusing in the sense that calreticulin is a gene and a protein which does many things, but when it was first found to be mutated in ET and myelofibrosis, it wasn't really clear that there was a link to JAK-STAT signaling. So let's talk about that a little bit more. So, 2013, two papers in New England Journal identified recurrent mutations in calreticulin in myeloproliferative diseases. And as shown with these pie charts here on the bottom, within polycythemia vera, we already knew that the vast majority of patients harbored mutations in JAK2, and calreticulin is essentially not present in that disease. But within ET and myelofibrosis, we knew that there was at least 40% or so of these patients that lacked the JAK2 mutation, and for a long time we just referred to those patients as JAK2 negative because we had no other alternative molecular marker other than the small sliver of patients that were known to carry MPL mutations. Now we know, based on these papers and several others since then, that calreticulin mutations are present in the majority of these JAK2 negative ET and myelofibrosis patients. And these mutations are quite fascinating. So they're invariably indels or insertions or deletions with a common end result, which is they result in a frame shift and the generation of a novel Cterminus. So the length of that can vary a little bit depending on the type of mutation, but they're very similar in this generation of this novel Cterminus and also concomitantly loss of the original Cterminus. So, when these mutations were discovered, the question was, well, what do they do? Could they activate JAK-STAT signaling? Again, it wasn't really clear at the time that there was a specific role for calreticulin in the JAK-STAT signaling axis. And very recently, this paper was published from Anne Mullally's lab in Cancer Discovery, and there are a handful of other papers that have been coming out also very recently reporting very similar findings, which are that the calreticulin mutation does seem to activate JAK-STAT signaling. That may not be the only mechanism by which it contributes to MPN pathogenesis, but it seems, based on these studies, that at least it's true that JAK-STAT signaling is activated by these calreticulin mutations. So to briefly talk about this study, in this paper, Anne's group first generated a mouse model with calreticulin, a retroviral transplant model, where they expressed the type 1 52 base pair deletion in calreticulin, which is the most commonly present mutation in patients with MPNs. And they found that expression of that mutant form of calreticulin was sufficient to induce an MPN phenotype. Specifically, these mice developed thrombocytosis and megakaryocyte hyperplasia. They also interestingly showed that if you express the calreticulin mutation in this BAF3 cell line, which is a classic cytokine-dependent cell line used in many of these types of studies, it results in cytokine-independent growth in the context of co-expression with the thrombopoietin receptor MPL, which would be similar to JAK2 V617F. So you can see the blue and red lines here. But it does not do that if the cells are expressing the EPO receptor or GCSF receptor, which is different from JAK2 V617F, which does induce cytokine independence. So it seems like these calreticulin mutations are specific to MPL, but they do not change things with EPO or GCSF receptor. They also showed in these cell lines that you could see activation of JAK-STAT signaling in cells expressing mutant calreticulin, but again, only in the presence of the MPL TPO receptor and not in the presence of these other type 1 cytokine receptors, EPO or GCSF. So again, specific to MPL, which makes sense in that you see these mutations in ET and myelofibrosis, but not in polycythemia vera. They also went on to show that that novel Cterminus that's generated by these calreticulin mutations, it's not necessarily about the specific amino acid changes, but rather that overall, this frame shift in novel Cterminus results in a change in the amino acid sort of charge status, which is that the frame shift results in the generation of a lot of positively charged amino acids. And you can actually replicate that simply by generating artificially a sequence that is mostly positively charged, but is not actually the same sequence that's present in these calreticulin mutations, and you get the same effect. So you see cytokine-independent growth with this positively charged mutant form of calreticulin, and you see activation of JAK-STAT signaling as well. So again, it's not the specific amino acids, but rather this overall net change to a positive region of amino acids. So let's see, I don't know how to go back. All right, so anyway, so again, these calreticulin mutations appear to activate JAK-STAT signaling and now can be grouped together with JAK2, MPL, et cetera, as being mutations that are part of this hallmark of JAK-STAT activation in the myeloproliferative diseases. So we're filling in the pie here now. You know, you can see that most of this pie is now colored, but there's still a fraction of those patients who would be what we call triple negative, meaning they lack JAK2, MPL, or calreticulin mutations. And also very recently, to further fill in that pie, work from two different groups identified what we might call atypical mutations in either MPL or JAK2 that are present in these so-called triple negative patients with myeloproliferative diseases. And in this one study here, they took a handful of these patients who were triple negative and performed whole exome sequencing to look for novel mutations. And what they found was that really the most striking finding was that in several of these patients, they could find alternative mutations in MPL in some of these patients that are in different locations than where the typical W515 mutations are, as well as one over here, W591. And I think these are color-coded because some of these are actually germline mutations. I think the blue ones are germline. The red ones are somatic. And I think this is an important point in that they showed functionally that there was perhaps a subtle functional effect from some of these mutations. Here looking at a reporter to look at JAK-STAT activation, some very mild differences compared with wild-type MPL. And in a growth assay, here's the MPL 515, which has a very clear phenotype in terms of cytokine-independent growth. These other mutations either had very little effect or a modest phenotype. This is the Y591. And so the functional effects, at least in this system, appear to be relatively mild. And the question is, are these really truly drivers? Are they maybe, do they need to cooperate with something else? Does it matter if they're germline versus somatic? And so it makes the clinical interpretation still not entirely clear as to whether these are really the full extent of what's causing these diseases. And the same is true with the JAK2 mutations identified. In this case, they were all germline, I believe, and the effects, again, were quite subtle functionally. So while it certainly makes sense that these could be the alternative drivers, it's not entirely clear whether these germline mutations are perhaps cooperating with other somatic mutations. But within these cohorts, they did not find any other recurrent mutations that made it clear what those cooperating mutations could be. So again, filling in the pie now, if you add in these atypical MPL or JAK2 mutations, the fraction becomes smaller where it's not clear, but this is particularly an issue still for those that are undefined. And so you can see within ET, of those patients that were triple negative, meaning lacking JAK2, MPL, or calreticulin mutations, you filled in a little bit here, but there's still actually quite a significant proportion within that group for which there are no clear driver mutations. And again, within ET, I think it's particularly an issue because to say that these are even truly clonal diseases in the absence of any molecular marker is very difficult. And so perhaps some of these patients aren't even clonal at all. And within myelofibrosis, that's probably less the case because it's more clear, at least based on the morphology of the bone marrow and things like that, that it truly is a clonal disease. But still, we're lacking a molecular sort of definition for some of these cases. Okay, so coming back to the germline variation aspect, so in 2009, it was reported in 3 different papers that there actually was a haplotype within the JAK2 gene, or JAK2 locus, that conferred an increased risk of MPN development. And this is just one of the examples from these studies which showed that the increased risk was on the order of around 2 to 4-fold for the specific JAK2 haplotype. And it actually, while at first it seemed, well, it's in the JAK2 locus and must specifically cause an increased risk of development of the specific JAK2 V617F mutation, but it turns out that this haplotype also, albeit a little bit less strongly, also increases the risk of MPN development, even if it's JAK2 negative. So it seems to confer a general risk of increased MPN development, but more so in the JAK2 positive cases. And it's thought that this is now responsible for about 50% of the population attributable risk of developing an MPN. And this has led to a lot of, I think, confusion in terms of what is actually going on here. The mechanism is not entirely clear still, but this is really thought to be contributing to the risk of development of sporadic MPNs and not these familial cases where there are multiple family members developed who have developed MPN. So this is in any person that there's an increased risk, 2 to 4-fold increased risk of developing an MPN. And the 50% risk over the population means it's actually quite prevalent in terms of that risk. So since then, a number of papers have come out looking at other possible contributors, and this is one example that was actually just published, I think, this week, and this was from Dr. Gottlieb and collaborators at 23andMe. And there are a couple of, I think, important and quite interesting points from this paper. So one is, the way the study was conducted was that patients with a self-reported history of an MPN sent in saliva samples to 23andMe, and then they were compared against a large number of population controls, so over 200,000, I think that were part of different 23andMe studies. And then they conducted a large GWAS study to look at different germline variants that might contribute to MPN risk. The first important point here is that, again, these were saliva samples, and this is the MPN patients, and these are the controls, and this is the JAK2 allele burden in the 2 populations. And you can see here, and this is stratified by age, so these are older patients in the controls, that actually there were a significant number of controls that had easily detectable JAK2 V617F. And so there are 2 points here. One is, first, saliva samples are heavily contaminated with blood cells. Otherwise, you should not be able to detect the JAK2 mutation unless it was germline. 2, the JAK2 mutation was present, again, in a significant proportion of the normal or the supposed normal population. And this now is well established through many studies over the past several years that the JAK2 and other mutations are present as part of this sort of clonal hematopoiesis of the elderly that Dr. Grauber touched on briefly in his talk. And so it might just be that these patients are carriers for JAK2. They might have clonal hematopoiesis, but they don't, in fact, necessarily have an MPN. So anyway, this was discovered in this cohort. And they then looked at the overall risk of MPN development, or more specifically also to look at other alleles that might contribute to an increased risk of MPN, or in fact just to carrier status for JAK2 V617F. And they found, when they looked across the genome, that this JAK2 haplotype that I mentioned earlier remained the strongest indicator or risk factor for development of MPN, but there were several others that seemed to be significantly associated with MPNs. And amongst those was a series of SNPs in the TERT locus, as well as within several other genes, including SH2B3, which is the same thing as LINC, and others, including CHEK2, which was rare, but also significantly associated as well with ATM, and those genes are involved in DNA damage, so that could perhaps possibly be the mechanism through which those contribute. So the point here, again, is that there seem to now be several germline variants that can increase the inherited risk for MPN development. And this is just another example where similar findings were reported. There are several other papers that have been coming out recently with similar findings. So we're now beginning to understand that it really is a combination of germline predisposition as well as somatic acquired mutations that contribute to MPN development. And so this is a schematic just kind of summing that up to indicate that what we've talked about primarily with the germline variation is here. The JAK2 haplotype is not rare, it's common. And again, it contributes to the risk in any patient of sporadic MPN development. As far as those sort of true familial MPN where there are multiple family members identified with MPN development, the real genetic basis for this is still not known. And then very rarely there are specific germline mutations in JAK2, MPL, or the EPO receptor which cause hereditary forms of thrombocytosis or erythrocytosis. Those are not necessarily really clonal MPNs, but really because they're germline, they result in a polyclonal increase in hemoglobin, platelet count, et cetera. Okay, so this I'm just putting up here. I know there's a talk tomorrow that's going to really elaborate on this, but just to say that a number of studies have looked at other genes beyond the JAK-STAT axis that are mutated in MPNs, and this is one example showing other mutations that are present, and these are mostly, many of these are also present in other myeloid malignancies. And again, this will be more a focus of a talk tomorrow, but also data implicating some of these genes affecting prognosis, specifically in myelofibrosis. But looking more broadly across the MPNs, this is probably the best example of a large exome sequencing study in the MPNs. And here are showing PV, ET, and myelofibrosis, about 40 patients in each group. And one of the points here I'll emphasize is that the number of mutations identified is different depending on the MPN subtype. So myelofibrosis, more mutations, about 13 is the median number. PV and ET less, so about 6.5. So the number of mutations differs. The spectrum of mutations is also different, although there's a lot of overlap. And I'm running out of time here, so I won't go over into too many details. But I'll make a couple points here, which is that the mutational profiles of MPNs versus AML, there's a lot of overlap, like I mentioned, but they are distinct. And so this is the spectrum of mutations in the MPNs, and this is within AML in 200 patients that were examined by the TCGA. And you can see that the most common mutations in de novo AML are in FLT3 and MPN1, and those are exceedingly rare in the chronic MPNs. Also, JAK2, MPL, and calreticulin are also extremely rare in de novo AML. And even within secondary AML from chronic MPNs, again, the mutational profile is very different than in de novo AML. Again, you see FLT3 only very rarely found in secondary AML from chronic MPN. Okay, and this is just a conceptual point that we think of these MPNs that they start out as a chronic phase, PV, ET, or myelofibrosis. They can go on to secondary AML. Quite interestingly, when secondary AML occurs, if a patient carries the JAK2 mutation of the chronic phase, it's actually fairly common that the AML that arises does not harbor the JAK2 mutation, which indicates that, one, JAK2 V617F may actually not be a strong driver of transformation, and two, that there must be some pre-JAK2 mutations that contribute to development or go on or ultimately are included in the clone that develop secondary AML. And I'm just going to skip over this part for the sake of time to get to another point. So as far as these so-called pre-JAK2 mutations, TET2 mutations were discovered in this paper in New England Journal as being present in some patients clearly acquired before JAK2. So it's thought, well, this, at least in some patients, is that so-called pre-JAK2 mutation. We know now, of course, that TET2 mutations are present across all myeloid malignancies, and they do not have to be acquired before JAK2. And that was further illustrated by this paper published in New England Journal last year from Tony Green's group, indicating that the actual order of mutation acquisition in the MPNs influences the phenotype. And this was focused on JAK2 and TET2. And what they did in the study is they grew out single-cell derived colonies from blood samples from MPN patients and then genotyped those for TET2 or JAK2 and identified a cohort of patients who harbored both mutations, but more importantly is that they were able to, on the basis of genotyping these individual colonies, identify patients who either had TET2 acquired first, and that was defined because they could identify colonies that only had the TET2 mutation. So therefore, you would, by inference, the TET2 mutation was acquired first, versus patients who had JAK2 only in some colonies, and therefore the JAK2 mutation was acquired first. And so they then looked at that in more detail, first with a group of patients, 12 in each group, who were either JAK2 first or TET2 first, and they found that those that were JAK2 first had a higher proportion of JAK2 homozygous mutant erythroid colonies, versus TET2 seemed to result in a proliferation of CMPs, chronic myeloid, CMPs, versus JAK2 seemed to be more slanted toward megakaryocyte erythroid progenitors. So they looked in an expanded cohort afterwards and found that when you look at colonies as well, if you grow out colonies from these TET2 versus JAK2 first patients, if you look at JAK2 first patients, you're more likely to find colonies that harbored both JAK2 and TET2, versus it was much less likely in the TET2 first patients to find double mutant colonies. And they basically showed that if you took colonies from these patients and looked at the gene expression profile, that they were distinct. Again, you could have patients who have the same two mutations, but the gene expression profile was distinct. And actually, they found that they are very different between the two groups. So again, same exact mutations, but very different gene expression profile. And clinically, they could show that there was a difference in terms of JAK2 first mutation. JAK2 first patients were younger at diagnosis and significantly more likely to be diagnosed with PV versus ET, as well that JAK2 first patients had a higher risk of thrombosis, and also in vitro, higher sensitivity to JAK2 inhibition. So it matters both biologically, and it seems to confer a difference in the clinical phenotype. And this is, perhaps the first actual clear description that the mutation order matters in any cancer at all. We'll skip that. So just to summarize, I hope I've impressed upon you that the disease phenotype in MPNs is really dictated by a complex mixture of germline and somatic alterations. It is clear that JAK-STAT activation is a hallmark of these diseases through a variety of different mutations, but there are a variety of other secondary mutations that likely modify the phenotype. And that clonal evolution and disease phenotype really can occur through a variety of different mechanisms, including the spectrum mutations, mutation order, et cetera, and this contributes to the extreme heterogeneity that we see in terms of the disease phenotype and progression. And I will stop there.